Bye. Okay, so good morning. We're going to talk about coronavirus in spite of all the problems, technical problems. I sent two links because this is a free version of the program. So the first one will be 40 minutes. And if any of you want to join and ask questions for the remainder, you can use the second link and we'll activate the second meeting and try to continue. So I don't think I really have to introduce the topic. So I'll just move to my next slide. Coronaviruses. So coronaviruses are actually positive strand RNA viruses. They're actually the largest positive strand RNA viruses that we know. Their genome is about 30 kilobases. I'll also say that I muted all of you, so you can unmute yourself in case you want to ask any questions. And I'll continue. We classify viruses both by their type of genetic material, which I said before, they are RNA viruses. And another classification that we use is if the virus is enveloped or not. It's an envelope that he gets from the plasma membrane when he leaves the cell. And you can see an electron microscope picture of the coronaviruses here. And you can see the virion particle itself and its envelope. And on that envelope, it has its spike proteins, which are the proteins that binds the receptor on the whole cell. And these spike proteins have a characteristic appearance that you can see here, and which resembles a crown, which is why these viruses are called coronaviruses. So we have hundreds of species of coronaviruses. They are mostly in animals, and only 7 are currently known to infect humans of these hundreds that I mentioned before. 4 of them actually cause just a common cold, and only 3 of them cause more serious diseases, as I'm going to show you in the next slide. So these are the coronaviruses that infect humans. Most of you probably remember the SARS epidemic at 2002 and 2004. This epidemic had 8,422 cases with a 10% fatality rates. It came from animals, as I mentioned before, as most coronaviruses do. And at some point, a close virus was found to that coronavirus that caused the first epidemic. It was found in bats, but it wasn't close enough to the variant that was found in humans. So it was assumed that the SARS had an intermediate animal. And I think my PowerPoint is stuck. Okay. And that animal was found in 2002 to be a civet, which is the animal you see here. That was the virus that was closest to the virus that was found in humans. And from those civets, it was found to transfer to humans. And then the next coronavirus that most of you probably haven't heard about, but there's an ongoing epidemic from 2012. It's called MERS, Middle Eastern Respiratory Syndrome, that you can see here. Most of you have not heard about because it's currently only in Arab countries, and I'll explain why in a second. We have 2,519 cases, and it's more causes more fatality cases than the SARS or the SARS-2 that we have now, 34.4%. The source is also thought to be bats, and from bats, it's probably the intermediate animal was identified, and it's actually camels, which is why you can see most of these cases in Arab countries. It hasn't gained the ability, we'll talk about how this happened in a minute, but it hasn't gained the ability to infect humans very efficiently. It's thought to be that each of the cases that we see is a new introduction from the animals to humans. And as I said before, it's pretty fatal. And we have the coronavirus that is causing the current pandemic, we can say now, which caused over 120,000 cases. The fatality rates, at least by the WHO, is 3.4%, and we'll talk about it in a minute. A close relative to this coronavirus was found also found in bats, but again, this bat virus is not close enough to the human virus. So again, it was thought there is an intermediate animal, and I'll talk about it. I'll show you the sequences in a couple of minutes. And at some point, the intermediate animal was thought to be a pangolin, which is the animal that you see here. We're not sure about that because the sequence of the pangolin coronavirus is close to the current SARS coronavirus 2 in a certain area of the sequence, but not all along the sequence of the virus, which, as I mentioned before, is a pretty long sequence. So we're not sure about that now, and I'll discuss that in a few minutes, and from there to humans. As you can see, bats are a major source of viruses, and it's currently a big issue in the field why bats carry so many viruses and why they don't get sick. There are a lot of theories about that. It's a very hot research topic. One assumption is that they have a higher body temperature when they fly, and that might be probably the reason why some viruses maybe are not capable to, maybe they're inactivated to some extent, and that's why they don't cause disease in those animals. There were also found differences in the immune system. Some innate immunity genes are elevated in these organisms, and maybe that causes low efficiency of replication. Okay, so that's about bats. So how do these viruses jump from animals to humans? So basically, as I mentioned before, coronaviruses have these spike proteins, and these are the proteins that binds receptors on the whole cell. The receptor for the SARS coronavirus 2 is known. It's called the ACE2 receptor, angiotensin converting enzyme 2. But basically, animal viruses have different spike proteins, and those spike proteins are generally bind animal cells, and they are not identical in sequence to the human ACE receptor. So basically, animal virus cannot efficiently infect a human cell. This can happen, but this is pretty rare. Sorry about that. This is pretty rare. And for that to happen, the virus has to change its sequence a little bit and gain the ability to efficiently infect humans. Once that happened, it has to undergo another step and to be able to transfer efficiently from one human being to the other. So these things actually happen from time to time, but because the virus doesn't efficiently infect humans, sometimes those are abortive infections, but sometimes they even manage to infect a certain human. He maybe feels some symptoms of cold, but then it doesn't efficiently transfer from human to human. So these events actually happen all the time, but they don't. Sometimes they don't develop into the disease in that certain person, or sometimes it's a mild disease and we don't even know that transfer occurred. So two things have to happen. The virus, the animal virus has to gain the ability to efficiently infect the human cell, and then it has to gain the ability to efficiently affect transfer from human to human. And those events are relatively rare. but they do happen. So how do these viruses change? So as you know, in our bodies, we have, when we replicate our genomes, we have the ability to correct. We have DNA repair systems. You can see on the x-axis the genome size, and on the y-axis you can see the mutation rate. And because we have a repair system, as you can see here, higher eukaryotes like us have, although we have a big genome, one of the largest genomes on this graph, we have a low mutation rate. But RNA viruses, or viruses in general, and RNA viruses in particular, they have their own RNA polymerases that replicates their genome, and that polymerases in general don't have an ability to repair themselves, which is the reason why viruses accumulate mutations. And they do that on purpose because they want these mutations to help them face all kinds of selections, such as medication or other selections that we impose upon them. Basically, coronaviruses are relatively stable, as I mentioned before. As I mentioned before, so I muted all of you again. So if you want to ask a question later, so please unmute yourself because all that noise bothers me. So basically, coronaviruses, as I said before, are one of the largest RNA viruses that we know. They do have some repairability, but they're still not as stable as our own genome with their repair mechanisms that I mentioned before. Okay, so let's look at the life cycle of this virus. RNA viruses in general have no life cycle step at the nucleus. All their life cycle occurs in the cytoplasm. So you can see here on the top, the SARS virus. It has the spike proteins that I mentioned before that actually bind to the receptor on the host cell and enable the entry of the virus. And the receptor, as I mentioned before, is the ACE2 receptor. Binding to the receptor induces endocytosis, as you can see here. And you can also see that once the virus enters the cell via the endosome, the viral membrane fuses with the endosome membrane, and the genome is released to the cytoplasm. This step of this fusion step, it was recently found that it requires some host proteases. I'll talk about it in a little while, but these proteases that are essential for this this fusion step and the maturation of the spike proteins is actually a good target for antivirals, and it was published in Cell a couple of days ago that one of the proteases responsible for this step has an inhibitor which is already clinically approved, and that is one of the not yet tried in clinical trial, but that's a good target and might be an efficient antiviral. So the genome is released into the cytoplasm, as you can see here, and most RNA viruses also translate their genome as a polyprotein, and they also carry proteases which cleave these polyproteins into the active proteins, as you can see here. So those viral proteases are also an attractive drug target, which we can talk about it later. As I said before, this is a very large virus. It has many proteins, and some of these proteins are translated later, transcribed later on the negative strand of this RNA virus. And we'll talk about the proteins in the next slide. After all the viral proteins are translated, the viral assembles in the cytoplasm of the host cell and is released so it can further infect new cells. as I said before, another, again, an important point that because all the replication is in the cytoplasm, these viruses carry their own RNA-dependent RNA polymerase, which is also an attractive drug target. We'll talk about it in a little bit. So as I said before, most RNA viruses that we know and that we work on have about 10 proteins. Again, this is one of the largest ones. You can see the list of the proteins here. And since the discovery of the SARS virus at 2002, nobody really worked on these SARS viruses too much. And as you can see, many of these proteins here, their activity is not known. There are also apparently two open reading frames that are different between the old SARS and the SARS-2. But there are a lot of proteins with an enzymatic activity, as you can see here, the exonuclease related to the repair activity, the RNA-dependent RNA polymerase that I mentioned before, the various proteases that cleave the virus, the polyprotein of the virus to the active proteins. So let's talk about a little bit about the let's talk a little bit about the spike protein of the virus. And I mentioned before that viruses can change, and that gives them the ability to infect human cells. So I want to show you some structural data from the old SARS virus, because it's, it's logically enough, it's more characterized than the current SARS. And it has this very nice illustrative slide. And then I'll talk a little bit about what we know about the sequence of the spike protein and its binding to the receptor with the current virus. I just want to show you how little mutations, a small number of mutations, can greatly change the affinity of these viruses to these cells. And that will maybe give you some understanding about how these infections actually occur. So on the left side here, you can see the human SARS receptor ACE2 and the human SARS spike from the 2002 and 2003 epidemic. And you can see that there's a large binding interface between these two proteins. Over here, one panel to the right, you can see the spike from the civet, which I said is an intermediate animal that transferred the virus to humans in the 2002-2003 epidemic. And as you can see over here, at least two mutations occurred. Two more mutations occurred, which I'm not showing here because they are not in this. They are not shown in this interface, but one mutation is a mutation into a lysin residue, which adds a charge here, and the other one also deleted a methyl group, which was fitting into this pocket. And these small number of mutations significantly affected the affinity of the viral protein to the human SARS receptor. So they found that this 4 amino acids difference causes a 1,000-fold difference in the affinity. So the civet spike protein is much less has a much less affinity to the human receptor, which then changed a little bit once it moved to human to create this much better fit that you can see on the left side. On the panel, on the next panel here, you can see the human ACE receptor on the top again, and on the bottom, a different isolate of the human SARS spike that also has, that was isolated a little bit later after the main epidemic, and you can see it has a proline here. And again, the deletion of the methyl group, which reduces the efficiency, and indeed it causes a smaller epidemic. And it was, in fact, it was much less efficient in infecting humans. And on the right here, you can see the rat SARS receptor and the human SARS spike. And you can see that the rat receptor has a large glycan group, and it lacks this hydrophobic pocket, which means it doesn't bind the human spike receptor very well, which means that, for example, that rats cannot be a very good model for studying SARS viruses. So let's see what we know about the current, what do we know about the spike protein of the current SARS-2 protein. You can see the whole virus up here, as I said, around 30 kb, and the spike protein is shown here. And we'll focus on two domains in that spike protein. One domain is the receptor binding domain that you can see here, and another domain which I'll discuss in a few minutes. And you can see here on the bottom the sequence of the closest viruses. The human SARS-2 protein is here, sequence is here on the top, and the closest bat virus is right under it, and then you can see the pangolin and the old SARS and another two related bat viruses sequences. And as you can see, there are 6 amino acid residues were identified. Six amino acid residues were identified to mediate the binding of the current SARS virus to its receptor. These residues are labeled in these blue boxes, as you can see here. And when you look at the bat virus, it has only one residue, which is identical over here, this L over here. If you look at the pangolin, you can see that all 6 residues, 6 out of 6, are identical to the current SARS coronavirus, which was the reason that people thought that the pangolin is the intermediate animal. But when they look at the whole virus, despite this high homology in the receptor binding domain, all along the virus itself, the homology is lower than the bat virus. So that's why this issue was questioned. There's another caveat to that, because the pangolins, the coronavirus that were identified from pangolins were identified from pangolins from an area which was not the area where the outbreak actually occurred. So one thought is that if they will sequence a pangolin from an area that is closer to Wuhan, they will can better confirm that the pangolins are really the intermediate animals in this case. And then there's a last residue that is sequenced here in the old human SARS, where a mutation occurred, this leucine to phenylalanine. Apparently that's one of the important residues for the interaction, but you can see that that residue is present also in some of the bat receptors. So apparently this residue alone is not sufficient, and it's probably some combination that is needed for the efficient binding. If you look here on the top, you can see that in the current SARS coronavirus, there is an insertion of this polybasic cleavage site that you can see here, this RRAR sequence. And apparently this is a cleavage site for a human protease called the furin, and that's absent from all of the coronaviruses we know. We know from other viruses like influenza that the presence of furin cleavage site at this location significantly increases the pathogenicity of this virus. So that's something that is new about the new coronavirus. This publication that that you can see here on the slide was published as an answer to people claiming that the new SARS was actually invented by the Chinese. and released from one of their labs as a biological weapon. And the claim against it in this publication that is referenced down here at the bottom of the slide is that in order to create, so if you do models for what's the perfect binding between the SARS and the receptor, you don't get actually this sequence of 6 amino acids. And if you want to try by chance to get this exact sequence, you'll have to test all the possibilities, which is 21 amino acids to the power of 6 if you have these 6 positions. So that's one piece of evidence that this was not originally planned. It probably occurs by a natural selection after various passages between animals and from them to humans. There's also another argument when you look at the sequence up here, this polybasic cleavage site insertion. So you would really have to know where to insert this polybasic cleavage site. And another argument against that is that proline residue that is right in front of the polybasic cleavage site, this proline residue enables the binding of various glycans that you can see here. And even if somebody would insert these polybasic cleavage site, you will probably not have thought of inserting this proline as well. The glycans are thought to protect the virus from the immune system in some manner to mask some epitopes. So, yeah, okay. So this is a recent study that was published in Science recently that was looking at the structure of the old SARS coronavirus versus the new one. And it found some interesting information that I want to share with you. In white, you see the old SARS coronavirus receptor binding domain, and in red, you see the residues that are actually different in the new SARS protein. And in this paper, they actually found that the new SARS protein is has a 10 to 20-fold higher affinity to the ACE2 receptor, which it shares with the old SARS coronavirus. Which means that, which might explain the fact that it's more infectious or spreads more rapidly. Another interesting thing that they show in this paper is that they took some of the old published monoclonal antibody against the first SARS virus, and unfortunately they did not bind this receptor at all. This has implications. If anybody thought of using old vaccines or antibodies that were developed from the previous SARS, they will probably not be helpful with this current protein because of the differences between these two proteins. Okay, so let's see if I can do this. I hope you probably all know this website by now. Let me see if I can share. I want to show you this website online. Let me see if I can do the technical thing needed to show you this online. Just a second. New sharing, probably here. New share, okay. Probably should be able to see that now. So this is a website that was developed by Johns Hopkins, and you can see here the total number of confirmed cases of the SARS infection, SARS coronavirus 2 infection in the world right now. This website has a lot of information and it's very helpful. For example, if you hit Italy, you can see here on the side the total number of deaths in Italy, the total number recovered. If you go to Iran or South Korea, which has more or less the same order of magnitude of deaths as infections, sorry, as Italy, which has 827 deaths. You can see that South Korea has only 66 death cases, and we'll discuss that in the next slide. So the death rates seem to be much lower in South Korea. We'll talk about it in a minute. Some other informative stuff, you can see here this graph that shows in orange the number of cases in China, in green the number of people that recovered, and in yellow the number of infections in locations outside China, which is right now 45,300. As you can see here, it seems that the number of infections outside China is, the graph is plateauing, but we have to see what happens in the next couple of days. You can see here this interesting jump in the number of cases in China, which took me a long time to find the reason. Apparently, up to this point, it was in the middle of the epidemic in China, and at some point they lost control. They weren't able to confirm all the cases using testing. So what they did is start reporting on clinically confirmed cases and not on laboratory confirmed. And back here, they managed again to test all the cases that they had. And as you see, this is the point that infections were starting to go down, and the graph, at least in China, plateaued. Let's try to go back to the presentation. And okay. Try to go back to the presentation. Let's see if I can do that. No, new share. Okay, back to my PowerPoint. Okay, here we go. Okay. Get this away. Should go back to the PowerPoint. No. Maybe like this. Share PowerPoint. I'm in a loop. Okay. Sorry about that. Share screen, PowerPoint. Okay, we're back to the PowerPoint. Okay, so I mentioned before that the death rates, the case fatality rates are different between the different countries, and that's a very interesting point. Death rates, case fatality rates are a little dynamic because some countries don't really know what's the number of actual people that are infected, because countries not all the countries test. Sorry about that. Okay, because not all the countries do actually adequate testing, and we'll talk about it in the next slide. Italy is one...
לא רואים את המקליט, יש בעיה בשיתוף של המסך.
לא רואים את המסך? אני עכשיו...
כן, לא רואים את המסך.
share PowerPoint, הנה. יש? יש?
רואים.
רואים?
אוקיי.
סבבה. אז אני אמשיך. Okay, so I'll repeat this slide because I understand you didn't see the screen. So what I said is that case fatality rates are different between the different countries.
לא רואים, אני חושב שכמעט כולם לא רואים.
לא רואים.
אתה רואה?
רגע, עכשיו צריך לראות. יש?
לא, כתוב שיש screen sharing, אבל לא רואים את המסך עצמו.
עדיין לא.
זה מוגדר כשארד אצלי. יכול להיות שעברו 40 דקות. מההתחלה של הסשן, ואז זה זרק אותנו.
בדיוק עברו, לא, חצי שעה עברו.
מ-10:30, אבל זה התחיל הסשן, אז לא בטוח ש... אתם עדיין לא רואים?
לא רואים.
רגע, בוא נעשה סטופ שייר, חכו, אני אעשה לך...
עכשיו זה בסדר.
עכשיו זה בסדר?
כן.
סטופ שייר, מאוד אינטואיטיבי. על הכיפק. אז אני ממשיכה, תגידו לי אם יש בעיות. אוקיי, אני חוזרת על הסלייד הזה שוב, אוקיי,
רואים רק אותך, לא רואים את ה... כן, לא רואים את הפאורפוינט שלו.
אז רגע, עוד פעם חדש, share, PowerPoint.
הנה
עכשיו זה
יופי, עכשיו. טוב.
Yes, okay. שלישית הסלייד. Okay, we'll do this again. So as I said, the case fatality rates vary from country to country, and there are several reasons for that. One reason is not all the countries test adequately, and I'll show you that in the next slide. So the case fatality rates are calculated as the number of fatality cases divided by the number of infected people, and that number will apparently change if the number of infected people is unknown or changes. The other reason is that different countries treated differently this epidemic and have different demographic characteristics. For example, Italy has one of the highest case fatality rates now, and one of the reasons for that is thought to be that Italy has a relatively old population, and I'll show you in a couple of slides that old people have a higher percentage of the old people get more severe disease, and old people have a higher percentage of fatalities. So all these things have to be taken into account. As I said before, it's very important to test because unless we test and we can confirm all these cases, we don't know really what's going on, and we don't know what measures we have to take in order to cope with this pandemic. As you can see here, Korea, with the lowest case fatality rates, did the largest number of tests, and that enabled it to identify the infected people and to properly isolate them and to reduce the amount of fatalities. As you can see, Israel is someplace here at the middle. True to March 8, we did around 3,500 tests. We're around 5,000 now. But for example, US has a big problem. They have, I'll talk about a little bit about it towards the end of my talk, but because a lot of Americans don't have a health insurance, and because they had problems with their tests, they only did 5 tests per million people, which is a very big problem because they don't really know the magnitude of the problem in the US, and they cannot isolate the contacts properly. But we'll talk about it in a few minutes a little more. So, how do you get infected with the coronavirus? Basically, the major route of infection is those droplets. There's a lot, I'll show you that in a minute. There's a lot of virus both in the nose and in the throat. There's almost we cannot find a virus in the blood. That's the major places that this virus replicates is in our respiratory system. And when we cough or sneeze, some viral particles are released, and if these will get into the airway or into the soft tissue of the person that is standing next to us, then that person might get infected. Depending on the physical characteristic, the weather, and many other things, you can determine the distance that these viruses are infectious at. And the SARS coronavirus 2 is more or less as infectious as Ebola. The Ministry of Health defined if you've been next to an infected person 2 meters or less for 15 minutes, then you have to be quarantined. So that's basically the numbers that we have now. We have other viruses that are infectious to greater distances. It was also found that some viruses shed in the stool. So at some point, people thought that maybe the fecal-oral route might also be relevant. But a study that were published a little later found that the virus isolated from stool was not infectious. So that's still a question mark. But the major route of infection is this airborne route. This is a study published in New England, recently published in New England Journal of Medicine. Basically, what I mentioned before, they tested by real-time PCR the amount of virus in nasal swab and in throat swabs. You can see the results here from 17 patients. You can see that right at the onset of symptoms, we have pretty high viral levels. there's more virus identified in the nose than in the throat. And this shedding pattern resembles influenza more than it resembles the old SARS, which was found to peak after around 10 days, and not like here, more or less around the onset of symptoms. And you can see that viral shedding through the nose and throat continues for about 12 or 15 days. It was also found in this paper that the viral load in asymptomatic patients was more or less similar to that of symptomatic patients, which indicates that asymptomatic patients can also transmit the disease. How does this virus spread so much and so fast? So we are to blame for that, our behavior. We live in a very crowded areas, and what you see here on the left is a small neighborhood in Beijing. As you can see here, it's very crowded, so infections can transfer from person to person pretty fast. And what you can see here on the right-hand side is the number of flights in 24 hours in Europe only. Which basically means that if we have viral infection someplace in the world, it takes a couple of hours until it can be transferred everywhere in the world. And that's one of the things that was banned, these flights, to try to stop the transfer of this virus from one end to the world to the other. But that's the reason we have infections spreading so fast around the world in this modern era. So how infectious is this virus? So basically, all of you heard that by now. We defined infectiousness by the average number of people that are infected from each person that is infected. This number is defined in epidemiology as R0. And as you can see here, SARS coronavirus is an infectious virus, but it's not the most infectious virus that we know. Measles is much more infectious. From every one person that is infected, 12 to 18 people get infected. And with our current SARS coronavirus, it seems that the average is around 2 people that get infected from each infected person. So it's not the most infectious virus that we know. It's also not the most fatal virus that we know. The fatality rates for Ebola, for example, are about between 40 to 70%, depends on the epidemic. So we're not in that bad shape. MERS, as I mentioned before, is around 34.4. Smallpox was around 30%. We think the World Health Organization published 3.4 figure for the current SARS coronavirus 2. But that number might go down if we identify, again, more infected people. So it's also not the most fatal virus that we know. And this is a table that shows both of the parameters I mentioned before, because it's important to look at both of them. If you infect a lot of people and the virus is not fatal, then you're still in good shape. So you got to look at these two parameters. And as you can see here, on the x-axis, you have the R0 number, average number of people affected by each sick person. And on the y-axis, you have the case fatality rates. And you can see okay. And you can see our coronavirus over here compared to other epidemics that we had before. And you can see that the situation is not good. We have to take it seriously, but it's not that bad. Coronavirus is somewhere down here with an average number of around 2.5 infected people per every person that is infected, and a fatality rate around 3.4% right now. Well, okay. So this is a slide that describes the first patient in the US. I show it because it shows a typical, more or less typical. There can be some changes. This looks at only one patient, but it shows nicely and graphically the development of the disease as a function of time. I'll show you in a minute another slide that looks at the different symptoms and how common they are. But this is generally what the patient feels during an infection. I have to mention that 81% of the infections are asymptomatic or mild. Only about 14% are severe, and the last 5 or 6% are critical. When you do get a severe disease, the severe disease occurs due to replication of the virus in the alveolar cells, which cause massive alveolar damage and progressive respiratory failure. In a regular mild form of the disease, the symptoms that most people will feel are basically fever that you can see here during most of the days of the disease, cough for the whole duration of the disease. Some will have rhinorrhea. Most of the people will feel fatigue. And then there is some minor reports. This is probably more rare of the involvement of the gastrointestinal system, but this is, seems to be a rare event, as you will see in the next slide. If the slide will decide to move. Okay, so here you see the symptoms of the coronavirus versus a common cold or flu. As you can see here, all of these diseases are relatively similar to each other, and it's very, very hard to distinguish. And this was one of the problems in Italy. They had a couple of patients suffering these symptoms. Some of them actually progressed to pneumonia, and they thought it's influenza. And by the time they actually discovered they have coronavirus cases, it was a little bit too late. So you can see here, fever is a common symptom, as well as cough. All the rest can occur sometimes. Diarrhea and runny nose are relatively rare. Again, these diseases are not much different in the disease itself, in the symptom of the mild disease itself than influenza. They are very different in the case fatality rates and the other parameters that we discussed before, and we can discuss a little bit later if you want. So let's look at the fatality rates by the different groups. This is a study that was published by the Chinese Center for Disease Control, according to the first 72,000 cases. Another study like this was published from the Koreans, which looks at less cases, and the numbers were more or less the same, except for one difference, which I'll tell you about in a minute. Basically males have a higher number of cases and a slightly higher death rate. You can see a breakdown by age here. And as you can see, mostly up to above 60-year-old or above 70 years old, most of the people will get mild or a severe disease, but the fatality rates are relatively low. And this has significantly changed, as you can see here, as you get older. The only difference in Korea was the 20-year-old group had many more cases, around 30%. We still don't know why that happens. You can also see here that children have no fatalities. There was a rumor that children don't get infected, but that's not correct. And a different study that was published recently showed that the percentage of infection in children is similar to the percentage of infection in the general population. So they do get infected. Though they probably get some form of milder disease. And another important issue regarding this disease, that if you have a pre-existing condition, you will have a much higher death rate, as you can see here, with the cardiovascular disease and of course respiratory disease being risk factors, as well as the other conditions that are shown in this slide. So one theory about why children get milder disease was recently published. And as I mentioned before, there are a few coronaviruses that cause common cold. So one hypothesis was that children are more infected with one of these common cold viruses, and that gives them a little bit of protection, of antibody protection to the current SARS virus. And this study looked at the fraction of children under 15 and the other age groups, as you can see here in this graph on the x-axis. And they found that indeed this cold coronavirus has a higher number of infection in younger people, and actually shows a reverse exactly a mirror image of the infections with the current coronaviruses, but that's only a hypothesis that is not proved yet. So, a few, about a couple of days ago, this article was published in a website called Stat News, which publishes scientific information. And it was an interview with an official from the WHO, which heads the emerging diseases unit. And it summarized, it debugs a couple of myths about coronavirus that were published in the recent days. And I want to go over some of these because they are really important. So first important fact is it seems that people are infectious early in the course of the disease. I showed you this before when we looked at the amount of virus in the throat and the nose. It seems that people shed for quite a while after they recover, but still it hasn't been shown that this virus shed late in the disease or after the person is recovered. It doesn't mean that this virus is infection. Sometimes viruses secreted or they're leftovers of RNA, but these viruses are not always infectious. So you don't have to get stressed about the fact that recovered people are infectious. In most cases, this is not true. So there was a big hype about the fact that asymptomatic people can infect others, and that was thought to be a major problem because we don't actually know that these people are sick, and if they can infect, we are in a big problem. But in this interview, the WHO claims that truly asymptomatic people are very rare. They mentioned a very small percentage of the cases. But again, this still, usually you don't test asymptomatic people. So these facts have to be tested more rigorously when we get to a point that we have time to test that. There was a myth that people are getting reinfected after recovery. This was debugged. There was a publication published a couple of days ago that shows that we have an antibody response and people are not getting reinfected after recovery. Those people who were published to have a virus after they were negative and released from the hospital, this was probably a problem with the tests. Transmission in China was mostly among family members and close contacts. Again, we mentioned that the fact that if we're crowded, you're less than 2 meters from a person, if you're not careful enough, especially in the same household. That was most of the transmission in China. We don't really know what is the contribution of community spread because when people are infected in the community, we're not sure from who they got infected from. This outbreak was not released by the Chinese. It's not driven by spread in the hospitals and not driven by spread in schools, especially due to the fact that children don't really get a serious disease. And again, we still don't know the true number of fatalities. I get asked a lot about pregnancy issues, and there was a study published a couple of days ago in the Lancet. This study looked at only at 9 people, so this information, when you assess this information, you need to take that into account. Basically, this study compared 9 pregnant women at the late stages of pregnancy that were infected with the coronavirus, and compared the clinical characteristic of their disease with the healthy woman. And apparently there was no difference. So pregnant women don't seem to get a more severe disease. And there's also no evidence for intrauterine infection caused by vertical transmission. All the fetuses from these 9 women were born without the virus. There is one report about a newborn child that after 30 hours had the virus, but we're not sure if he probably got infected after he was born. Why does it take so long to make a vaccine? So I don't need to explain that because you're all biologists, but basically the main target for vaccine is the spike protein, which is on the envelope, and this is actually what our immune system can see. And basically what you want to do is you want to create antibodies that neutralize the binding of the spike protein to the receptor in order to neutralize the virus. And there are various attempts to create such vaccines. All of the vaccine strategies that we know, people are actually trying to utilize them right now, starting from RNA vaccine, which you heard about from a company called Moderna. They basically insert RNA of the spike protein and let that spike protein be translated inside our body, and hopefully that would create antibodies. There are DNA vaccine expressing the spike protein. People trying to express recombinant spike proteins and inject them to the body. We have vectors that express the spike protein. People are trying to make activated and live attenuated vaccines. All these are under research and development at various stages, but you have to remember that you're all biologists, you know how difficult it is to do these things. And they might work and might not. And even if you successfully create the RNA or the vector, you have to make sure that it elicits an immune response and neutralizing antibodies. And even after you do that, you have to go through all of the clinical phases. You can't skip the safety tests, and each of those takes a while. You first start with a small number of patients, and then you go to a larger number, and you cannot skip any of these steps. And even if we have this vaccine, it will have to be mass-produced. That, as you know, even this upscaling can take time, and the errors can occur. So, as you know, it's not simple in biology to upscale things. So, the time frame is really, really unclear, and it's going to take at least a couple of months, even despite the massive efforts that are given to this. And I truly hope I'm wrong, and we will have a vaccine very, very soon. What might help us in the meantime is these drugs that are being developed. So basically, the major approach is to take drugs that are already approved for other indications, or for example, other viruses, the previous SARS, Ebola, and to try to take these drugs that already passed some of the development steps and some of the safety steps, and to try to use these already approved drugs and test them against the SARS coronavirus 2. There are tons of tests that are clinical trials that are being conducted now in China and around the world. The major targets, as I mentioned before, the proteases that are involved in the maturation of the spike protein, the RNA-dependent RNA polymerase. Most of the antivirals that we have today are RNA-dependent RNA polymerase inhibitors, nucleoside or nucleotide inhibitors, and also protease inhibitors that are targeted against the viral protease, which is responsible for cleaving the viral polyprotein into the active virus proteins. And as I mentioned before, this new chemostat mesilat is the inhibitor that was found to inhibit one of the proteases. It's a clinically approved drug for a different indication. It was published in Cell a couple of days ago that it can inhibit one of the proteases that is responsible for the maturation of the spike protein. Other important targets, other important drugs is that you hear about a lot is this drug that you can see here, produced by Gilead, Remdesivir. This is a polymerase inhibitor that was developed against Ebola, and it seems to have some clinical efficiency against the SARS protein, and it's been tested right now. You can see the other drugs listed here. I only put in this table drugs that are in advanced stages of the clinical trials, as you can see here. And as I mentioned before, most of them are RNA-dependent RNA polymerase inhibitors or protease inhibitors. Hopefully, some of them, there's one drug that was previously known as a drug for malaria, which inhibits endosome acidification and blocks the entry of the virus. All of these are in clinical trials. None of these was approved yet. Hopefully, these will will be approved soon, and can at least help us alleviate some of the severe cases of this disease. So what's going to happen? So there's been a lot of press announcement that 70%, 60% of the world population will be infected. Basically, these assumptions are built on mathematical models. Some of the experts that publish these assumptions, these models, actually corrected some of their assumptions to a lower range now. They say between 60 to 20% of the people around the world will get infected. Basically, what these people did, they extrapolated from what happened from viruses with a similar R0 virus values, like the pandemic flu in 1968. And they actually used what happened in China to build mathematical models. But we have to remember that, of course, this might be true, but you have to remember that these are mathematical models, and they are based on assumptions, and these assumptions might be different or might change. I put on this slide some things that could change this assumption. For example, for models that are based on what happened in Wuhan, these conditions in Wuhan might be different in other countries for some reason. These models are built on the R0 values that we have now, but this could be wrong because there are more or less people infected. We still don't know the exact numbers. The controls measures that we're taking might give us some time to prepare. Seasonal factors might affect the outcome, and we'll talk about it in the next slide. We might be able to identify a vaccine, and that, of course, will change the course of the disease. Will coronavirus go away in warmer weather? This is an excellent question. So basically, this assumption started from other cold viruses that we know, and what happened from the 2003 SARS virus that actually disappeared. And what we know from other common cold or influenza viruses. But again, we cannot know for sure about this virus. This virus is new. And the most important point about this virus is that compared to influenza or other viruses that are in the population now, we have, against influenza and against other viruses, we have some sort of immunity in the population. We get infected again with influenza because the virus changed, but still we have some cross-immunity from previous influenza infections that we have. Some of us are vaccinated, some of us experience various forms of this disease. The problem with this coronavirus is that we don't really have any immunity in the population against this virus. And because of that, we cannot really predict what this virus is going to do. Regarding influenza, it was found that the virus favors dry and cold condition. We don't know that about the SARS. People are more indoors in the winter, so maybe they get more readily infected. That might change in the summer. A lot of people say that there's no influenza in the summer, but that's also not really true. There is influenza, but at smaller amounts. So really can't count on this issue to significantly affect. We don't know. We hope it will, but we cannot really count on this to significantly affect this. Masks, yes, no, maybe. So basically, a surgical mask is supposed to protect the patient and not the person who wears it. So it's a good idea, this mask, if you're sick, not to infect other people. But it's not good enough to protect yourself. For that, there is a different kind of mask called the respirator. But generally, most of the health organizations, such as the CDC or World Health Organization, don't recommend masks to people in the community. They recommend masks for people who are working in a healthcare setting. Because for many reasons, if you cough or sneeze, then you probably will take the mask off, so you can infect people anyway. Most of the people that bought these masks bought one mask, and basically these masks have to be changed several times in order to remain efficient. And there are other, the pores in these masks are 0.3 micrometers. The virus is smaller, so it can block viruses that come in large droplets, but it probably will not block the virus itself. So this is not a recommendation for me, do as you wish, but basically the general population, even when they wear a respirator, it's not completely sealed. So basically all the various organizations do not recommend a mask for healthy people. They recommend a mask for sick people in order to protect them from infecting others. What we can do, so basically you should wash your hands, do everything that, all the recommendations from the Ministry of Health, disinfect your area, avoid touching your face, don't travel to infected areas, don't eat or touch wild animals, isolate patients. That's basically, you all know that by now. I want to show you how administrations cope with these epidemics. Basically, there are 3 stages. Some administrations publish this in various versions. I took this from the Singapore Ministry of Health. The first step is the alert step. You hear that there is an epidemic somewhere else around the world. So basically, you want to try to minimize the importation of the disease and stop the spread from individual cases by isolating them and by tracing their contacts. The next stage is containment of the disease. And basically, you do that by tracing the various contacts and quarantining them. And then when you cannot do that anymore, what you basically do is try to mitigate the spread to reduce the overall impact of the disease. And the idea behind this mitigation policy is what you see here. This graph was in the media. Basically, if you don't take any protection measures, you have this large peak of disease, and that will exceed the health system capacity. So basically, the idea behind all these isolation and vegetation measures is to reduce the number of cases and make sure they occur during a longer period of time, and that will enable the healthcare system to cope with these cases, which, especially in Israel, with our healthcare system that is already saturated, would be very important, and this is what I assume the Ministry of Health is trying to do right now. The timing of interventions is very important. the previous graph is actually the goal of what you want to do. You want to flatten the curve, but how do we know that it's work? I'll show you two examples. One of them you see here is what, this is a very good example, although it's from 1918, influenza pandemic. You can see here two scenarios. One is Philadelphia and one is St. Louis. Philadelphia had the cases earlier, but they actually ignored them and didn't take any social distancing measures. They actually had a parade right after the first cases that you can see here to celebrate the end of World War I, and that caused this peak of cases that you can see here. In contrast, St. Louis, sorry about that. In contrast, St. Louis took extensive measures of social distances, and they closed the schools, canceled all the parades, and you can see here that the curve was flattened. I also wanted to show you an example of how this happened in China, but this seminar is already taking too long, so I don't want to take more of your time, and I'll skip this. I also wanted to show you what's going on in the US right now. You know, I'll show you, if any of you want, you can just log out. That's the advantage of a virtual, and I can show you this slide. So feel free to log out. I won't take this personally, and I'll keep on talking. So basically what you can see here is another example of how social distancing measures work from what happened in China. Basically in orange you see the daily official number of cases, and in gray you see the true daily coronavirus cases. And you can see the true cases started around December 8th. The Chinese didn't even know that this is a coronavirus 2. It was not identified yet. And there were a number of cases of unidentified, unusual pneumonia. The virus was identified at January 7th. Once the virus was identified, a couple of days later, they were able to test for this virus, and then you can start seeing the official cases going up. Basically, around January 23, they started to isolate patients and started locking down the Wuhan area. And as you can see here, when they did that, they knew they were able to test, and they knew only about 400 official cases, but the true number was much higher. And when they did that, when they did this lockdown, the number of true cases immediately started dropping. We still see the official cases going up because there was more, people were more aware about this disease, and they started going and seeking care. But this number also starts going down following. So it seems that these social distancing measures have some effect. I want to show you an interesting thing, and what we can now, not me, but what people can do now with sequencing regarding what happened in the US. And you can see the source down here. It's from a blog published by a person from a project called NextSec in the US, which sequences now many pathogen samples and shares them. So they have all the coronavirus sequencing now, and they can build these evolutionary trees. But I just want to show you a small part of it and how we can learn from this sequencing about what's going on in the community, and we can learn about this disease. So the first case in the Seattle area happened in January 15. And as you can see, when they sequenced the virus, they found that it's completely identical to isolate from China, which indicates that this first case in Washington originated from China. 5 weeks later, they had the second case, which was a community case from an unknown source. And when they sequenced it, they found there were only 3 additional mutations from the sequence they identified in January 15. And when they looked at the rate of mutations in other isolates of the X virus, they found that it has about 2 mutations in 1 month. So they concluded that this was the same insertion. This is actually an identical sequence with only 3 additional mutations that occurred in these 5 weeks. So basically what this means is that in Washington and Seattle, they had virus spreading in the community for 5 weeks they weren't aware about. And basically, that meant that they actually had a lot more cases than what they knew at that point. And this actually came true when they started testing, and you can see that on the graph on the right. So, as I mentioned before, one of the problems that caused that is that in the US, because people don't have any health insurance, they did very little testing, only 5 tests per million people. And it wasn't only the health insurance problem, they also had other, and they still have other issues with their tests. One of that is that they had actually a this slide speaks to biologists, they actually had a contamination in their PCR, their negative control was showing a result. And now they solved that, and the CDC distributed the good kits, but they still have a problem. Now they seem to be out of RNA extraction kits in order to perform the PCR. So the US has serious issues with this pandemic, and hopefully they can overcome them before it is too late. And this is my last slide. We talk about coronavirus a lot, and coronavirus is not the flu. It seems to be a more severe disease, and the fatality rates seem to be higher. But still, we are very freaked out about 120 cases, 120,000 cases as we should be, because there's no immunization in the community. But you have to remember that for influenza, we have this slide describes only this last winter season, this last flu season in the US. And in the US only, we have over 34 million influenza cases and 20,000 deaths. And we do have a vaccine, but most of us don't go